Hi, I'm Nicholas, a PhD student in computer science using high-performance computing to help design better pneumococcal vaccines. This video focuses less on the computational details. Instead, it gives a brief overview of the problem we're tackling and how computers help us solve it. Our story begins with Streptococcus pneumoniae, a bacterium responsible for hundreds of thousands of deaths every year. For a long time, our tools to fight back were limited. That was, until the development of pneumococcal conjugate vaccines. These vaccines protect us by training the immune system to produce antibodies that recognize and destroy S. pneumoniae. So, the next time the bacteria tries to infect you, those antibodies are ready to fight back. Let's take a closer look at this process. On the surface of the bacterium is a capsule, a thick layer of sugars. Zooming in further, we see the individual sugar molecules that form this capsule, known as capsular polysaccharides. The capsular polysaccharide, or CPS, is important because it's where antibodies bind when they attack the bacterium. Understanding this process is the focus of my research. However, it has been made more complicated with the discovery that there isn't just one type of Streptococcus pneumoniae, but more than 100 different types, each with their own unique CPS. Unfortunately, antibodies that protect against one type usually don't work against another. This matters because both our bodies and vaccines are incredibly complex. Creating a vaccine that protects against every type of S. pneumonia is nearly impossible. The vaccine would simply have too many components. This means we have to be smart. We start by targeting the most deadly and common types. And, if we're even smarter, we can take advantage of something called cross-protection, when an antibody protects against not just one type of S. pneumonia, but two or even more. In other words, by adding just one component to the vaccine, we can protect against multiple types. This makes the vaccine simpler and more affordable. So, how do computers fit into all of this? Well, I use high-performance computing to study the capsular polysaccharide, how it interacts with antibodies, and when cross-protection might occur, helping inform vaccine design. Using powerful computer clusters and physics-based models, we can simulate the motion of every atom in these systems on a picosecond timescale. This allows us to explore the behavior of the CPS and its interactions with antibodies in far greater detail than is possible in a physical chemistry lab. After much time and computation, we can hopefully uncover the mechanisms behind CPS antibody interactions, insights that can guide smarter vaccine design and deepen our understanding of a system central to human health. If this kind of research interests you, check out our group's publications, visit my poster at the School of IT Showcase, or reach out to me at my email here to learn more about my work.